Hello, everyone, and welcome to this pre-meeting institute on an introduction to the neurobiology of traumatic stress. My name is Jasmeet, and I'm actually speaking to you from Boston today because unfortunately I wasn't able to get out to LA because of Hurricane Sandy. So this TMI will not proceed exactly as planned because two of us will be bringing our presentations to you via teleconference instead of in person. However, the good news is that you'll still hear from all of the speakers who were originally scheduled to speak, and the last three speakers will be there in person. And just as a warning, there appears to be a 5 to 10-second delay in advancing my slides. So I'll try to stay on target, but someone will notify me if things start getting a little bit out of sync. Okay, so here is the schedule for today. Hopefully you can see that. So the first three items on the schedule will be via teleconference, so my talk as well as Dr. Shin's talk will be via teleconference. However, Dr. Milad's talk and Dr. Rasmusson's talk and Dr. Amstadter's talk will be there in person. So this workshop today is set up as a series of lectures providing an overview of the state-of-the-art in different neurobiological methodologies used to study PTSD, and particularly for those not familiar with these literatures. So just to introduce you to the speakers and the talks, I'm a staff psychologist in the National Center for PTSD and assistant professor at Boston University School of Medicine. So I'm going to be providing you with an introduction to fMRI, essentially what it measures, what it takes to collect fMRI data, and some limitations of this method. And that will segue into Dr. Shin's talk on brain abnormalities of PTSD and Dr. Milad's talk on neuroimaging PTSD. Dr. Shin is professor and chair of psychology at Tufts University, and her work in applying fMRI to study PTSD has been instrumental in uncovering the neural circuitry of the disorder. And Dr. Milad is associate professor at Harvard Medical School and has conducted some of the groundbreaking work examining fear conditioning and extinction in PTSD using fMRI. Following Dr. Milad's talk, we'll have a 20-minute break, and then afterwards you'll hear from Dr. Rasmusson, also of the National Center for PTSD and associate professor at Boston University School of Medicine. Dr. Rasmusson's work has contributed to the current expanded view of the neurobiological factors that contribute to PTSD risk and stress resilience. And finally, we're fortunate to have Dr. Ananda Amstadter, assistant professor at Virginia Commonwealth University, and her work has made important discoveries regarding the genetics of PTSD, depression, and substance use disorders. So after each talk, we will reserve 5 minutes at the end of each talk for you to ask questions. And if you don't have time to ask your question within the allotted time, please feel free to contact me afterwards, and I can get you set up with the person who can answer your question. So why do we want to present a PMI on the neurobiology of traumatic stress? It's important to study neurobiology because ultimately neurological and psychiatric disorders arise from the result of integrated actions of brain networks. And these actions are enormously complex and operate at many different levels, from the molecular level and genetics to the level of thousands of neurons collectively working as measured by fMRI. So by studying how the molecular and brain networks function, we can better understand disorders themselves as well as discover ways to treat them. And so this is a particularly exciting time to study neurobiology because advances in technology are occurring at such a rapid pace. Hopefully you can see my slide here, just giving the example of neuroimaging. In 1990, there were no papers using fMRI because fMRI really had not been discovered yet as a tool to understand brain function. But by 2006, as this figure shows, there were over 80,000 citations per year for just fMRI alone. But you can see there's also a rapid increase in citations for PET and SPECT imaging, as well as MEG and EEG. And so neuroimaging has provided some of the strongest evidence for brain networks involved in PTSD, as you'll hear more about from Dr. Shin and Milad. Genetic technologies are also rapidly occurring. As an example, a recent study that just came out about a month ago identified the Aurora gene as being associated with the presence of PTSD in a genome-wide association study, which is really an exciting discovery in that it can lead to new insights to the neuroprotective mechanisms of the brain. So studying the neurobiology of stress has yielded some pretty important discoveries. However, given all of that, it's still important to understand that these methods are not without limitations, and many of them are based on mechanisms that we have not yet fully characterized and don't quite understand yet. So therefore, these methods, for the most part, are still relevant for research, but with some exceptions, are not yet ready for prime-time clinical application. And so in today's talk, we'll not only review the exciting discoveries made in recent years, but also a summary of the limitations of each method. Okay, so let's move on to an introduction to fMRI. This is in no way a comprehensive overview, but the purpose of this is just so that you have a better understanding of what goes into the studies that Lisa and Mohammed are going to present, basically the assumptions that those studies make and how the data are collected and analyzed. Was someone going to say something? Okay. So before I go into...
Jasmeet, if you could speak up a little more, some of the people are having difficulty hearing you.
Okay, I'll try. All right, I'm moving closer to the phone. Okay, so before I go into detail about fMRI specifically, I want to mention that it's just one type of imaging you can do using an MRI machine. You can also do structural imaging, which looks at, for example, the volume or shape of different structures of the brain. So some of the most important results in PTSD have come from brain volume studies, and specifically a structure called the hippocampus. And with MRI, you can also look at white matter of the brain, doing diffusion tensor imaging studies. And there are other types of imaging methods as well, but these three methods, fMRI, structural MRI, and diffusion tensor imaging, have been the three methods that are the main ones used to study PTSD. Okay, so now turning to fMRI. As opposed to structural MRI, functional MRI measures functional activity related to task performance or the brain at rest. And results from fMRI studies can show regions that are involved in a particular function. And as you'll hear from Dr. Shin and Dr. Milad's talk, regions that seem to be altered in PTSD. fMRI is a technique for measuring metabolic correlates of neuronal activity. And this is an important point because fMRI does not directly measure neural activity, like, for example, single unit cell recording. Rather, it's based on the BOLD signal, which stands for blood oxygenation level dependent signal, which I'll explain more in the next few slides. Okay, I'm just going to wait a second to make sure my slides caught up to where I am. So hopefully you can now see this slide called fMRI in bold. So this figure shows the entire pathway from a neuron's activity all the way to fMRI brain data. So first,